Hello, everybody. My name is Adrian Chavez, and I am very happy to welcome you for today's video presentation. As we know, we have been talking about these past weeks about herbivore interactions, and this week's paper helped us on having a better idea on how plants can have their defensive mechanisms. Well, so you might see some familiar faces in the picture down there in the corner of the slide. That's Dr. Phil's hand laboratory in Gainesville. And actually, we have been reading one of his papers that they published this year, which help us to have a better idea on how plant chemical mechanisms can deter herbivory behavior. But it's also this also help us to see how these insects have their own mechanisms to overcome these plant defensive mechanisms, and that's the case. Just to point out, insects can have detoxification mechanisms which will help them to detoxify harmful compounds, and also there are some other mechanisms like sequestration, which they refer to in this paper. Well, before going further with the reading, I would like to break down first our main characters in today's reading. On the picture on the left, we have Dioscorea bulbifera, also known as air potato, which is an invasive species here, and it was reported since the early 1900s. On the middle, we have Lilioceris sheni, and on the right side of the slide, we have Lilioceris ejena, which are two biocontrol agents that can help us to reduce the invasive consequences of the air potato. These consequences have been already reported. Invasive species can provoke severe damage, not just to agriculture, but also to natural ecosystems. And that's the case just here in the US, where losses up to $120 billion were reported because of invasive species. But here we don't talk just about insects or plants. Here we talk about large animals, many other organisms like viruses, bacteria, and many others. And to control these, the biocontrol agents Lilioceris sheni and Lilioceris ejena and many others are very important. As I mentioned before, some insects can have sequestration mechanisms as their counterattack for plant defense. And that's the case of Lilioceris sheni, which has this sequestration mechanism that allows it to extract from the plant the chemical compounds, and he will later use them as mechanisms to avoid being eaten by predators. But unfortunately, that's not the case of Lilioceris ejena, which is a non-sequestering herbivore, but it can metabolize the natural compounds of the plant, but he will not use them as a mechanism of defense against other predators. And before going deeper on the reading, I would like to point out our two also main characters for today's reading. On the left side, we have the jasmonic acid, and on the right side, we have salicylic acid. Well, on the left side, what does jasmonic acid does in the plant? Its role is mainly to activate some responses whenever chewing herbivores are feeding on the plants. And this will help the plant to activate some secondary metabolites and also some protein inhibitors that will help to reduce insect feeding, and it will also affect the insect performance. The salicylic acid also plays an important role on plant defense, and this will help to strengthen the cells of the plant, and that will delay the growth of the pathogen. So, with this background information, I would like to go back to the paper and try to break down step by step on what the authors did. So, to see how chemical compounds applied to the air potato affects the herbivory behavior, the authors have the two hypotheses. The first one, Lilioceris ejena will feed more and perform better on bulbils that were treated with JA and plants that were previously damaged by Lilioceris sheni. And the second hypothesis is that both species, L. ejena and L. sheni, will show hump-shaped responses to increased concentrations of diosgenin at low, intermediate concentrations, and at high concentrations. So what the authors did to test the first hypothesis was to use this assay with 4 different treatments. The first treatment was to apply JA on an air potato plant. The second one was to apply SA. The third control was the water control check, and a fourth treatment that was an air potato plant infested with L. cherny beetles. After 7 months of the applications of the different treatments, from each of the treatments, bulbils were harvested, and then each of the bulbils were sliced and were offered as a source food for larvae of L. ejena. And from these experiments, the data was collected. So what they collected was the frass weight, the wax weight to measure the total of the bulbil that was consumed, the pupal weight, and also the beetle weight. So after all the time that they wait for collecting the data, we have some important results. If we talk about the panel A and B on the graph, we can see that there was no significant difference between the treatments and the control. If we talk about the panel A, we can see that the amount of food that the larvae was consuming did not differ between the treatments. But then, if we look into panel C and D of the graph, we can see how the salicylic acid did have an effect if we compare it to the control and to the other two treatments. The pupae that came up in this panel C was heavier if compared to the other treatments and the control. And this trend is also what we can see in the panel D of the picture, where we can see that the adult weight was also better if compared to the other treatments and the control. So, meaning that all the treatments didn't change the feeding amount that the larvae was consuming, but the quality of the food did change the insect performance. So, to better understand how much the larvae was eating and how this was affecting their growth, the authors also ran different statistical analyses. Here they look at frass and wax weight as measures of food consumption and also pupal weight as a measure of performance. And by comparing different treatments, they tested whether eating more was having a greater impact on the pupae or whether the relationship changed under the different treatments. And that's what we can see in the graph. Here we can see what the larvae was eating translated into its growth. And we can see that in the control and the beetle treatment, the more they ate, the larger the pupil was. But as we saw previously, the jasmonic acid didn't lead to a bigger pupae, which means that the food quality was lower. And this is the opposite in the salicylic acid treatment, where eating more had a better impact on the pupae growth, meaning that it actually improved the food of quality for the larvae. Okay, after finishing for testing and collecting data for the first hypothesis, they also did have a second hypothesis that was to see different levels of diosgenin and see what was the responses of the beetles to these levels of concentration. Diosgenin is also a natural compound that occurs in several Dioscorera species, and here they used different concentrations in the dose-dependent PSA. So what the authors did for this was that they used adult beetles that were placed inside deli containers with diet discs, and each of the beetles received only one concentration of the treatment. And what they collected from here was the frost weight to see the feeding of the beetles and also the survival after 10 days. What they also did was that they initially the weights of the beetles were recorded to have a standardized comparisons. Okay, and from this part of the experiment, we also have some interesting results. As we can see here, they will help us to have a better idea on how these two insects were having different answers to the plant defensive mechanisms. And if we look into the panel A of the graph, we can see how Lilioceris sheni was performing good at the medium level of the treatment, and then it declines with higher concentrations of the treatment. And this trend is also what we can see in panel B of the graph, where we can see how the survival of Lilioceris sheni was good at the intermediate levels of the treatment, but then it starts to decrease at higher concentrations of diosgenin. So from these A and B panels, we can have an idea that L. escheni tolerates and can also benefit from the moderate levels of the treatments. But if it has higher concentrations, it will not play a good role on the insect. Okay, and now if we look into the left part of the graph, panel C, for the other species, we can see how this species was answering to the different treatments. And opposite to what the other species was doing, here we can see when the concentrations of the treatment were higher, the feeding activity of this species was less, meaning that a higher concentration, less feeding activity. And this trend is also what we can see on panel D, where we can see how the survival declines at higher concentrations of the different treatments. So from this, we can strengthen the idea that this insect was not a sequestering type of insect, meaning that higher concentrations levels of the treatment, it will decline its survival, meaning that this insect will prefer not to eat on this plant, and then with thus it will have lower survival. So overall, this graph show us and highlights how the two different species can have different strategies to deal with the plant defensive mechanism. So to start wrapping up today's discussion video, I would like to point out some of the ideas that came to my mind after reading the paper, and this is also what the author highlights and make emphasis on, is that both beetles can inflict damage in additive way to the plant, meaning that it will not affect each other's feeding performance. That's something very interesting and important to take away, and also that the amount of the food that the larvae was eating didn't change much across the treatments, but what was making the difference was the quality of the food that they were eating, and this was having an impact on their growth. And also that the treatment JA can reduce the quality of the food, but this will not also affect that much the feeding behavior of the insect. And what the authors also emphasize is that with this experiment, that previous damage of the herbivory and also the application of plant hormones to the leaves of the air potato will not impact the overall feeding of the beetle Lilioceris echena, but it will lightly influence its performance. Okay, with these two main ideas, I would like to finish this part of the video. As the authors mentioned, and we knew through the paper, these two species don't feed on the same part of the plant. That's why they can not compete with each other, but together these two species can put a stronger pressure on the invasive herb potato. And it's also possible to use more than one agent for biocontrol, but before this, we should know how their interactions with plant defense are so that we understand how they can work together. Okay, and before going further and proposing the questions, I would like to suggest these two readings. The first paper is also from Dr. Hans' laboratory, and in here they conducted a field survey between 13 different sites across Florida. And here they find out that the JA was increasing the feeding and preference by the beetle Lilioceris sheni. So they had a previous idea on how this was affecting the feeding behavior of this specialist beetle. And the second reading I would like to recommend, it's about how the invasive species have been causing economic damage here in the US in different levels. Okay, and these are the three questions that I would like to propose to continue with this discussion. And with that, I want to thank you all for your time and your attention, and I look forward to seeing your answers on today's discussion.